Good morning, everyone. I think we can start now. It's almost 9:10, but I think we can start now. Let me share first the agenda of today's agenda. Okay. Good morning, everyone. Thank you very much for coming today. Good morning, Prof. Wayan. Good morning, Dr. Channarong Rodkhum. And good morning, Dr. Dini Wahyukartikasari. And good morning, Dr. Nuning. Thank you very much. Good morning, everyone. Thank you very much for coming and in online lecture series 2, Molecular Technology and One Health. This is the second series of the online lecture, and this activity actually is the collaborations between One Health Collaborating Center Universitas Gadjah Mada and the Biotechnology Program, Graduate School Universitas Gadjah Mada. Thank you very much. And as we already know that today we will learn about the molecular technology, about the method, especially in the zoonotic disease and also in the aquaculture. And also we will relate it with the One Health approach that we also learned from previous meeting, what is One Health and everything. And we will learn more about it today. And then, as we already know about the agenda, I already show you in the screen that this activity will be opened by the head of doctor study program for biotechnology, and then continued with the first speaker, Prof. Wayan Tunas Artama, and then the second speaker is Dr. Channarong Rodkhum, and then we will continue with the question and answer section and to closing. Okay, without any further ado, I will invite Dr. Tririni Nuriring Tias. Good morning, Dr. Nuning.
Good morning.
Okay. Yeah, the screen is yours.
Okay, thank you very much, Mbak Putu. These guest speakers, Prof. Wayan and Dr. Channarong Rodkhum, and then all participants, ladies and gentlemen, and also my dear friend of science, technology, and innovation. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Well, I'm pleased here to welcome you to the second biotechnology webinar series. I'm so glad that we are all now gathered here. And first, allow me to greet again our valuable speaker today, Dr. Channarong Rodkhum from Chulalongkorn University, Thailand. Thank you, Dr. Channa. And then Prof. Wayan Tunas Artama, our own professor here from One Health Collaborating Center UGM. Thank you, terima kasih Pak Wayan atas kesediaannya menjadi membagikan sharing experience-nya Thanks a lot also to Dr. Dini Wahyukartikasari who are willing to be our moderator today to guide this webinar series. Well, I also want to greet some of my friends here. I saw in the list of participants, I recognize some familiar name there. Ada Pak Anto, and also then Bu Sylvie and other friends. Thank you very much for joining us, and also our colleague and alumni from Biotechnology Study Program. Thank you very much for joining us today. I hope this webinar series will become way to use to always keep in touch, and also maybe our next speaker also can come from our alumni actually. So I'm sure that it will be very interesting. So the biotechnology webinar series is actually our routine agenda. Sorry. Okay. And to organize it, the Biotechnology Study Program are collaborating with One Health Collaborating Center and also Research Center for Biotechnology UGM. This agenda is actually meant for promoting and also sharing the benefit and the important role of biotechnology as well as molecular biology to support and to solve the problem in the community. Through this webinar series, through this webinar series, we hope that we can distribute and then also to introduce different kind of biotechnology approaches, and then we can utilize it to support and maybe then to change the world. Yeah. Today's topic are very relevant with the situation that we encounter recently. Yeah, Prof. Wayan will talk about how we can develop the detection kit, yeah, from zoonosis disease, yeah, and then also Dr. Chana, yeah, I think, and then also here. From Chulalongkorn, yeah, we have also we will talk about Dr. Channarong will talk about aquaculture, yeah, about the resistancy, antimicrobial resistant monitoring. So I'm sure that those field is also very important because we can find antibiotic resistant that in many fields, yeah, it can be also from aquaculture, livestock, and also agriculture. So I'm sure we can learn a lot today from our two speakers. So I will not take a lot of time. Yeah, so I'm sure that all of you already enthusiastic and cannot wait for the talk. So I wish you all a very successful webinar and thank you very much for your joining. So thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Nuning. Yes, I agree with you that I hope that from today's session, all of the participants can get a lot of insight and they can learn from our speakers' experience. And maybe we can start the collaboration between the participants.
Yeah.
That would be a great result from us. Thank you very much once again, Dr. Nuning. And I don't want to spend more time for the participants and the speakers. That's why I will move the session to the main session, the lecture session. But before we start it, let me invite Dr. Dinisari and introduce to all of you about her CV or her profile. Wait a minute. Okay. Okay. So this is Dr. Dini Wahyu Kartikasari, SBIMSI. She is a head of master's degree program for biotechnology, Graduate School of Universitas Gadjah Mada. And she is a member of the Biotechnology Research Center UGM. And she is a member of the Genetic and Fish Breeding Laboratory, Fisheries Department and Agriculture Faculty, Universitas Gadjah Mada. She finished her Ph.D. about gene regulation research in gene regulation research laboratory, biological science in Nara Institute of Science and Technology, Japan. She finished her master degree in biotechnology, Graduate School of Universitas Gadjah Mada, and her bachelor studies come from the aquaculture, fisheries department, agriculture faculty, Universitas Gadjah Mada. And she have published a lot of international and national publication regarding about the related about the fish and other research. Without any further ado, I would like to invite Bu Dini or Dr. Dini. Good morning, Dr. Dini.
Good morning, Mbak Putu. Good morning, everybody, ladies and gentlemen.
Yeah, thank you very much for coming. I will give the screen to you. Thank you very much.
Yes. Thank you, Mbak Putu. So from now, I will lead the discussion about diseases in zoonotic and also the molecular method, the sophisticated molecular method that we will discuss today from two incredible our guest speakers today. So before all of before the talk, I would like to read some housekeeping items. So in this webinar, participants may ask the speakers during the course through a pigeonhole at molecular 2. And then other issues may be asked in the chat room. And then all of the participants will get e-certificate. E-certificate will be shared a week after the lecture, and the speaker's material will be shared with the speaker's permission, of course. And then we will share the link for attendance at 10 minutes prior to the end of today's course. So you will add some your information. So from there, then we will send e-certificate and also material that shared by our speakers today. Next. So today there are two talks from Professor Wayan Tunas Artama and also from Dr. Channarong Rodkhum from Chulalongkorn University. And first from Prof. Wayan. Prof. Wayan is sorry. Could you please make a bigger, Mbak Putu?
Is it okay now?
Yes. Okay. Enough.
Sorry.
Okay. So Prof. Wayan graduated from Faculty of Veterinary Medicine, Gadjah Mada University at 1978. Then he graduated from doctoral degree in biochemistry for Veterinary Institute, Freie Universität Berlin, Germany in 1999. Currently, he is a permanent lecturer at the Faculty of Veterinary Medicine, Universitas Gadjah Mada, and also coordinator of One Health Collaborating Center, Universitas Gadjah Mada. He was the Deputy Dean of Academic Research and Community Services at the Faculty of Veterinary Medicine and was awarded the British Council Research Award. He is actively conducting research on toxoplasmosis and writing articles both in national and international journals. He has appeared in several national and international seminars. He has been conducted many activities in One Health. So Dr. Y, and then the second speakers will be Dr. Channarong Rodkhum, Ph.D. Dr. Channarong. Okay, so we will proceed from the first talk from Professor Wayan Tunas Artama first. Professor Wayan, you will have a 40-minute talk, and then we will proceed for the next speaker then. Okay, Prof. Wayan, time is yours.
Okay, thank you, Budini.
Thank you.
First of all, I would like to thank the organizing committee and also Dr. Dini, Dr. Nuning to make today's discussion available. And also, I would like to thank Dr. Channarong Rodkhum, DPM, Ph.D. from Chulalongkorn University. Thank you for your participation. May I share my topic? Yeah. So, anyone see my? Sorry.
Yes, clearly.
Yeah. I want to share again from beginning.
Okay.
Wow. Sorry. Okay. Good morning, everyone.
Good morning.
I would like to share. My topic is the application of molecular technology in zoonotic disease detection for human risk reduction. This is connecting One Health, zoonotic disease, and biotechnology with the 2 points that nowadays is very important in the worldwide because of in the last decade. And we facing a lot of problem with the zoonotic disease as well as with emerging infectious diseases. And then I try to connect the One Health.
Itu pertama dalam umur hidup itu Pak, pergi ke tempat begitu.
Bocor. Connect One Health with biotechnology. What is the role of biotechnology in the today's advanced technology? Then help us a lot with the RDT, rapid diagnostic test, and also NGS. And I want to talk for today. My lecture topics, I decided in 3 points. One is regarding the zoonotic disease trend. What is the zoonotic disease in today's? And the second one is the public health response and One Health. What is One Health and how we should respond with the zoonotic diseases? And then how we use a new technology like RDT, microbiome analysis, NGS, and helping us a lot for controlling the zoonotic disease as well as emerging infectious diseases. This is 3 major points that are connecting zoonotic disease with biotechnology. So biological confirmation for biotechnology approach. The interesting of biotechnology is the use of biotechnology in this today problem is how we can confirm the disease, the pathogen that is causing the disease, this biological confirmation. But it's not easy. It's because of need a lot of equipment and also the manpower and also the facilities. Therefore, we try to connect the efficiency of using molecular tool to detect and to control how to control the zoonotic diseases, emerging infectious disease. This is my point here is molecular test is the key because according to the today's situation in COVID-19, rabies, and Ebola, MERS-CoV infection, and we should have tool how to detect virus variant and also how to identify the cases and how to control the cases and to do control the zoonotic diseases. This is the point that I want to stress for this my talk today. So many problems, we call that a wicked problem. There's a lot of problems never completely solved, like what we have in the previous
ada kejadian bisnis ini selalu ditemukan.
In the last decade that we have it's H5N1, H5, and with the avian influenza. So from the beginning to 1983 in Indonesia until now, we still have the disease spread from one province to another province, and now we have 30 provinces in Indonesia. Then 33 still only one is free from avian influenza of bird flu. So that's way we have problem is never solved completely because due to the system farming in Indonesia, because we have here system farming is different with others in developing countries, because like a backyard farming is very difficult to control, and therefore it's not possible and not feasible or not possible to vaccinate all the chicken or chicken, and because of not easy to get it. But in the good farming system, like in the first farming, like in the industrial one, it's easy because to control is easy. Biosecurity and biosafety is easier than in a backyard problem. So then every problem is unique, like what we have now, COVID-19 is very unique, and then the problem is the definition is everybody have a different perspective, and this is difficult to, and also the multiple stakeholder, not only the veterinarian physician, but also need social sciences and any other discipline that should work together. So every solution need a system how to the control, how to delivery to address this problem to the will. This is the problem of today's that we have. So then we have challenges ahead. The facts, like in the 21st century now, many infectious diseases regarding the zoonosis and emerging infectious disease increase more than we predict. Zoonotic infection are those of non-human origin. In fact, most zoonotic infection is come from wildlife. So around 75% of the human diseases, human zoonotic diseases is come from wildlife origin. Or in the tail, that is around 62 of human diseases are animal origin. So the first line, animal diseases, is very important. How to we handle with the human animal diseases, not spill over for the human being. This is our challenge ahead. And then the government, international WHO, have already packaged action package, 16 action package that in order to control the zoonotic disease. Like they have a program on, we call it PREVENT, for antimicrobial resistance and zoonotic diseases, biosafety and biosecurity, and also immunization, and the DETECT program, so national laboratory system constructing and real-time surveillance. We do an active surveillance and supporting workforce development and respond, and how we should respond for the outbreak, emergency operation center, and linking public health with a multi-sector rapid response and medical encouragement, personal development, and so on. So through this program, detect, identify, detect, prevent, and respond. This is a core of how to control the disease in our world today. So talking about zoonotic diseases, as in my introduction, is zoonotic diseases is spread between animal and people. So the definition of this is the disease that naturally transmitted from animal to human being, or vice versa, from human to animals, and around 75% of the emerging zoonotic disease emerging infectious diseases, and why and how the disease is spread from animal to the human could be through direct contact, indirect contact, through vector-borne disease, and foodborne disease or waterborne diseases. There are many pathogens for zoonotic diseases. I just mentioned here some of the pathogens that really have a big problem for the human population. So pathogens are there bacteria, parasite, viruses, fungi, and others. So just an example, bacteria like salmonellosis, brucellosis, leptospirosis that we have focused also on our research project on leptospirosis, and for parasite diseases we have also our project on toxoplasmosis. And then for viruses, this is many viruses appear in the last decade. Rabies still have that big problem in Indonesia and also in Asian countries. Bird flu we have now, still have now since 1983, and Ebola in West Africa, but unfortunately we don't have Ebola here in our region in Asia, and fungi. This is okay. How the disease transmit from animal to the human? We call it spillover transmission mode. Direct contact that I mentioned before, like many different diseases that I already mentioned in the last many years. There's like tularensis, and many other diseases, rabies from saliva. This is direct contact. And then contaminated food, we still have a salmonellosis problem even in meat or egg. Leptospirosis, especially for farmer, we have a many time outbreak in Indonesia, especially in Yogyakarta. We have 3 times outbreak of leptospirosis in 2010. Then until now, we still have a problem with leptospirosis, and many provinces in Indonesia reporting that they have leptospirosis problem. For vector-borne disease, like Rift Valley fever, equine encephalitis, and Japanese encephalitis through mosquito and malaria, of course. Also for tick. And then last, we have a problem with the reservoir, like bats and rat as a reservoir for many different diseases. Bats is a reservoir for SARS, MERS-CoV-2, Ebola, and SARS-CoV-2. This is the biggest transmitted the disease from animal to the human being. Like we have a SARS problem, and then we have MERS-CoV, and we have Ebola and SARS-CoV-2 for COVID-19. They predicted this come from wildlife, also from bats. So talking about the emerging infectious disease, we have in the last decade a lot of disease reported. That the red one is a newly emerging infectious disease, like 2009 they have H1N1, and now we have Ebola, and still have Ebola in African countries, and we have COVID-19 in worldwide. 215 countries have a problem with COVID-19, and then re-emerging infectious disease like rabies, Lissa virus, and we have still problem with this disease. So, okay, more than 200 emerging infectious diseases now reported from WHO. So for the emerging infectious disease, I would like to introduce for all of you that how did the disease will accelerate from time to time. So we know that the connection between wildlife disease, livestock disease, and human disease. This is the to accelerate the disease transmitted or spill over from animal to human or vice versa. Is connection with the climate change. Climate change is one of the drivers, but mobilization of people, population is the most problem, because the people can move from one place to another places in a very short time, and then transmitted the disease from a country to countries. Like we have a pandemic now, and then increase the agriculture trade and wildlife trade also a problem, and rapid urbanization and deforestation, of course, intensive farming system because of the population increase, and then we need more food, and then increase the farming system. So that when then we need one health approach. Yeah, one health is the interface of the ecosystem health, animal health, and human health. But in general, this is need a transdisciplinary approach. Many different disciplines should work together to handle and to control the disease, zoonotic diseases. So for One Health is very important, and how the One Health connect to the new technology is very important to control the problem of the zoonotic disease, because zoonotic disease is a very complex problem. So therefore, the One Health for everybody that already have knowledge on medical science in, related to human disease, animal disease, and environmental problem. That is many different disciplines involved in the One Health. Social sciences, of course, and then medical, veterinarian, ecologists, and this because of a problem that we facing day to day because of food problem and food safety, that a lot of people eating is food from wildlife, and of course that's very dangerous for the people. We don't know what is kind of the diseases over there, and then IMR and antimicrobial misuse is also now become a problem, and a factor, of course, for the certain diseases, and water pollution that we have everywhere, and deforestation. So One Health is this allow us to work across sectoral collaboration between many different sector. That the core of the One Health, we should have a knowledge, hard skill of the One Health, and then we should have soft skill of One Health, like collaboration, management, because this disease is very complex problem. Then we should have a collaboration among others stakeholder. So I put just in one example of COVID-19. How should we handle COVID-19? That we have cooperation. The 3 point, we call that 3C, but we start from cooperation. So if we have a problem with COVID-19, it's not easy because a lot of problem there. including many different disciplines and many different problems, social as well as medical problem. So we should have a coordination among the stakeholders. We should have a good communication. And then if we have a good communication, coordination, and then we should have collaboration to managing the disease. What should we do? So every region have a different characteristic. So we should have belief the culture, like our soft skill of One Health, believe on culture belief that every region have a different character, different culture. We should respect for their culture. And then for the scientists and from medical and from veterinarian, they should identify is there new variant of the viruses, and then how should we control these diseases, this COVID-19. So we should have also research on surveillance, that testing, tracing, and also surveillance, active surveillance, as well as in human and also in animal. So this one, all sectors should work together. So we call that in Bahasa Indonesia, we should collaboration in the planning and also in the preparation and also the evaluation of managing COVID-19. So start from this outbreak, we should identify which the disease, where is the disease, and when, why that even over there, and how should we control, how we should control, and then we should integrated approach we do, and then for biotechnology, we should have a new tool for molecular technique, then will be addressed by the second speaker later on. Yes, I want to introduce what is the important one in controlling zoonotic disease, that molecular technique is very important. Then we should have a regional cooperation among others countries, and we should have an integrated project together, and international cooperation, of course, because we need vaccination, RDT, rapid diagnostic test for antigen test, and also for antibody tests. So the first line, we should have for controlling emerging infectious disease, we, like as I mentioned before, which of the disease and what is the disease. For example, COVID-19, when is this happen, where the disease is, and how should we control it. This is, we call it a system thinking. We should have a system thinking to control the problem. So then we do need a solution for that. Now this internet-based information technologies is very important. Improve the disease reporting, because if we do good reporting or standard reporting, international reporting system, then we understand the progress of the disease and how we should control, and how to facilitate the emergency communication and dissemination of this information for the government and for the community. Then we said that this is the connection of biotechnology and public health, and we should have an analysis of genome, maybe the variant of virus because of RNA virus mutated very fast, and then we should identify the variant and microbiome maybe analysis, and metagenomic analysis is the one problem to solve this procedure. So the role of human genetic disease susceptibility, progression, host response, and microbiome metagenomic analysis is one of the important thing to do. So my popular diagram here is from emerging threat program. Firstly, we should identify the pathogen. We do longitudinal surveillance of wildlife, domestic animal, to identify and characterize a specific microbe that might cause serious disease in human. This need a biological tool to identify the pathogen and to do a good active surveillance. And then we can understand, we do understand the risk determination. We can characterize the risk that microbe might possibly to human, its transmission route from animal to human. And after we know that risk determination, and then we can prepare for the response. What should we do to outbreak response? Strengthen country capacities. We train people. We have facilitated the laboratory. We should have communication among the government and the laboratory. We should have a laboratory network. We should have a good communication, and then we prepare for the response program. Then we have human risk reduction to minimize the disease spillover from from animal to human, or vice versa, could possibly also from animal from human to animal. That's why we have a project related to that program to analyzing microbiome and maybe spillover of the disease from animal to the human. We have a project on bats, long-tailed macaque, and also for dog. This will be a spillover and amplification of the disease. So this on another one is our favorite subject. If we do forecasting readiness yearly from wildlife, this green one is a wildlife. If we have a early detection procedure, early detection tool, maybe through RDT, rapid diagnostic test, through molecular diagnostic, and then we can reduce the spillover, minimize the spillover from wildlife to the animal, livestock, and then to human. If you saw here in this picture, this is if we don't have a control, we don't have a rapid diagnostic test, we don't have a forecasting readiness, and the problem will be very high. But then if we have a control program, and then will be reduced like a small one here, in human as well, from white to be the red one. So this is the control benefit if we have done One Health approach to managing the outbreak, disease outbreak. The last topic, the last session, my topic, we introduce all of you how important is biotechnology in managing zoonotic diseases or emerging infectious diseases. So biotechnology have many different subjects, many different disciplines, increase the capacity of diagnostic, and we have here nanotechnology, we have infrastructure in management, and then for information, they develop many things, also biological computing through bioinformatics. We connecting the human disease and biological and computer and also mathematics. Then we can use this in the next step for next generation sequencing. So we have also in biotechnology that develop also a tool like early diagnostic system through molecular approach. This is just an example developed through the intensive research in biotechnology that the company can develop very easy to detect the disease using immunochromatography. For that we understand with the antigen test for COVID-19 and antigen for many different diseases, and also antibody detection to know our response, the body response to the disease. And then at the beginning we have antibody kit for detecting COVID-19, for detecting SARS-CoV-2 virus, and now we have antigen detection and also PCR-based detection. So many different RDT that's already developed, and also we can find in the market. For bacteria, many different kit already developed through biotechnology industry, like for detection of Clostridium perfringens, Corynebacterium, Leptospirosis, and microbiome TB, and also Neisseria meningitis, Salmonella TB, Vibrio cholera. For viruses that we have now a lot of RDT for virus like adenovirus, rotavirus, influenza virus, dengue virus, SARS-CoV-2 virus, Lissa virus, and Ebola virus. And for parasite, many different parasite already easy to detect using the rapid diagnostic tests like Plasmodium, Giardia, and Cryptosporidium. So I use this technology to map the disease in Middle Java because we have a parasitic disease surveillance in many different provinces, in Bali, in Yogyakarta, Middle Java, and West Java. Then we have a result here to show to all of you that the disease transmitted distributed. We using a rapid diagnostic test to identify and to map the disease. This is in regional in West in Middle Java, and here we get a very good result that the disease is contribute. The disease was we can find the disease along the river around 200 300 meters from the river side. The disease spread to that region, and then using a GPS we identify the disease even in the area that highland area. We identify, we collecting sample more than the district here, but we don't have a problem with toxoplasmosis in that area. But in the area where the river we get positive for the population over there. So talking for the molecular technique, and this technique will maybe later more explain from the second speaker, from our colleagues from Chulalongkorn. This is just, I just mentioned that in 1977, just the beginning of the DNA sequencing. At that time, the first technique that for sequencing technique established by Dr. Sanger, Prof. Sanger, and then in 1990 become molecular of the year 1990 this PCR by Karen Mollis, and then human genome in 2005 start the 1995 until 2005 the human genome already sequenced. Then they introduced the first NGS over there, and now a lot of pathogen genome, human genome, bovine genome already established using the new technology, the latest technology that we have also introduced how to have to identify the pathogen easily using a small mini ion in our laboratory, but not yet in operation, but just we established in biotechnology. We do have a small new latest technology for the sequencing procedure. So this is just an example how to from the mix sample we can extract the DNA, and then we can do a shotgun metagenomic sequencing, and then we have a long read, and then we can identify the sample using metagenomic sequencing, and then we can get the sequence of the bacteria. So this is just the new technology that just we got from our colleagues, from our collaboration with our project with GSPS project. We established now in the biotechnology, but not yet running well. But we still have to train people and to establish the laboratory in the biotechnology research center. This is a mini ion nanopore. It's very simple. It looks like one small equipment, but we can do a long read in less than 24 hours. Maybe you can ask our colleague that already done this in Thailand after that. So the nanopore or mini ion is very important to detect the species detection. We can do species detection, and we can also doing strain typing, and even antibiotic resistant profile. We can do that using Oxford nanopore mini ion sequencer. So just connecting like our endpoint, connecting to the PC or laptop, and then let it done in less than 24 hours. We can get what we want to analyze. Just as an example that I was done before, we can identify bacteria from the genus until the subspecies of bacteria using mini ion sequencer for bacterial identification. Okay, maybe this is my last picture. I want to share if you have any time to go to Yogyakarta to the Biotechnology Research Center, we established the mini ion center over there. Okay, I think this is the tool that connected biotechnology. How is the important biotechnology tool, molecular technique for for detecting pathogen like a zoonotic pathogen or emerging infectious pathogen. Okay, thank you. This is my last conclusion. To prevent the disease, we should have a strong program to prevent movement of pathogen and vector via modern transportation. This is very important one. The spreading of the disease from one country or from one place to another places because of pathogen or vector through modern transportation. And second one, improve international cooperation and data sharing to anticipate the fatality rate of the disease and to control the disease, and then improve effective laboratory-based surveillance. We should have an active surveillance for identify the disease in the region. And the last one, rebuild public health infrastructure to prevent and control vector-borne and zoonotic diseases. Like we should have train the personnel how to train the people for the zoonotic disease and maybe potential disease in their own region, and then laboratory epidemiological capacity tool for mosquito control, vaccine, understanding the disease ecology. This is also very important. The last one, political will. This is very important also because the disease regional prevention and control program is very important. The country or the province should have their own regional program to control the disease and to map the disease, and then economic support, of course, for, and we should have apply One Health Smart, One Health system mapping analysis resource toolkit, that we map the disease in their own region, and then we train the people in the region, and then how to anticipate the potential outbreak in the region. Okay, I think this is all my talk for today. Thank you so much. This is wildlife. Okay, thank you, Dr. Dini.
Yes, thank you so much, Prof. Wayan, for the very nice talk. So, Prof. Wayan talk is very close related with our situation today about zoonotic diseases around us today about SARS-CoV, and the very important thing is molecular identification is a key for early detection for human risk reduction. That's very important. And before we proceed for the next speakers, I remind you, all of the participants, you can write your question and also comment in the pigeonhole that already shared in the chat room. So you can put your discussion over there. And then for the next speakers, we are our welcoming second speakers, Dr. Channarong Rodkhum from Chulalongkorn University. Are you ready, Dr. Channarong? Yes, Dr. Channarong Rodkhum, DVM Ph.D. Maybe some of you, what is DVM? Asking what is DVM? DVM is Doctoral Veterinary Medicine. Is that correct, Prof. Wayan? So Dr. Channarong Rodkhum,
Right.
Right. Okay. Thank you. So Dr. Channarong Rodkhum graduated from Faculty of Veterinary Science, Chulalongkorn University for his DVM degree and Graduate School of Marine Science and Technology, Tokyo, University of Marine Science and Technology, Tokyo, Japan. His research is emphasized on fish infectious diseases and pathogenesis, especially bacterial and viral pathogen of fish such as some bacterial and also some virus. The current project is development of nano delivery system vaccine for potentially use for prevention of bacterial diseases outbreak in tilapia and Asia sea bass. He has published many international journal and national. So Prof. Channarong Rodkhum, time is yours. Thank you.
For Dr. Channarong, he will use the video, Udini, so I will play the video for him.
Okay.
Good morning, everyone. My name is Channarong Rodkhum. Today, I would like to talk with you about the molecular research in Thailand's aquaculture, pathogen characterization, and antimicrobial resistance monitoring. First of all, I would like to thank you, One Health Collaborating Center, Universitas Gadjah Mada, Indonesia, for inviting me to be a speaker today. I come from the Center of Excellence in Fish Infectious Diseases, Department of Veterinary Microbiology, Faculty of Vet Science, Chulalongkorn University, Bangkok, Thailand. Before I talk about the molecular research, I would like to introduce you about the aquaculture in the world and in Thailand. Right now, to meet the world seafood need, aquaculture production need to increase by 46.4 million metric tons, and is dramatically increased every year, year by year. An estimate 73 to 180 billion fish are farmed each year, making them the most farmed vertebrate in the world. Higher fish welfare mean better life for billions of fish on farm. For Thailand, yield from freshwater aquaculture by species from Department of Fishery Thailand. You can see that in Thailand, we have the main species, including Nile tilapia and red tilapia. Another species also high production in Thailand, also such as walking catfish, around 27.4%, and the striped catfish, around 5.7%. For world production of tilapia is about 1.5 million tons per year, the most consumed fish globally, and it is now called aquatic chicken. For successful and sustainable in fish farming come from the successful in fish health management that have to be composed of many factors, including health booster, such as the probiotics, immunostimulants, vaccines, farm water quality, and environmental management, breed, such as the disease-resistant fish, biosecurity, farming skill, and basic knowledge and nutrition. If you would like to have a successful and sustainable in fish farming, you have to concern about many factors, composed of many factors, not one by one factor. Then you will have a successful in fish farming. For the fish health management, we have to have the appropriate stock density, appropriate nutrition for each breed and period, water quality management and monitoring. Sometimes we have to supplement with immunostimulants such as vitamin, trace mineral, and beta-glucan. We can use the probiotics or symbiotics for promote fish health. Finally, we can use the vaccination for prevention the important infectious disease, both bacteria, virus. The failure of fish health management is mean the farm will have many diseases occur for fish, both infectious disease and non-infectious disease. Today we will talk about only the infectious disease because we would like to talk about the molecular research about the pathogen caused the infectious disease in aquaculture and or in fish. The important infectious disease of tilapia including streptococcosis, columnaris, hemorrhagic septicemia such as the Aeromonas septicemia and Edwardsella septicemia, Franciscellosis, tilapin virus infection, or somehow we can call TILV or tilapia leg virus infection, parasitic infections such as Trichodina and Gylodactylus. Right now, we have some emerging viral disease such as the tilapia parvovirus also. Finally, the disease that usually occur in tilapia is not come one by one. It's usually appear as the mixed infection or concurrent infection. For the tilapia culture, we can separate into many phases of culture, start from hatchery, nursery, pre-grow out, and grow out. You can see that in almost all period of tilapia culture, we have some infectious disease that important for each period of culture, such as Flowobacterium columnar, Sapolegnia, and other fungi, Trichodina, Dactylogerus parasite, Rickettsia-like organisms, Iridovirus, and the Streptococcosis or Streptococcus species infection. This picture shows the clinical signs of streptococcosis in tilapia. It will be including the exophthalmos, or you can call pop eye. You can see the erratic swimming of fish because the bacteria is infect brain of the fish or infect meninges of the fish. So it will show clinical signs of erratic swimming. And another clinical sign, you can see hemorrhage around the base of fin and some area on the body surface. Neck disease is columnaris disease. Is cause high mortality in fish, especially the small fish in the hatchery or in the growing period. The clinical signs that you can see is the necrosis around fins or some area of the skin, and the obvious clinical signs that you can see in many tilapia fish is the gill necrosis lesion. If fish has the necrosis at the gill like this, it's difficult to survive because gill is very important organ for fish. Next disease is the bacterial hemorrhagic septicemia caused by many bacteria such as Edwardsella or Aeromonas species. You can see the big area of hemorrhage on the skin fin of the fish. And when you make a necropsy, you can find hemorrhage in the internal organ. Next disease is Franciscellosis caused by Francisella species, especially Francisella nortunensis. In Thailand, you can find the white spot nodule in internal organ of the fish like this. Next disease is the disease caused by virus we call tilapin virus infection. You can see the clinical signs such as hemorrhage at the skin and fin rot, tail rot, and also you can see the big belly of tilapia fish. When we make a necropsy, we can find the ascitic fluid in abdomen of the fish. And when you do the histopathology, you see the histopathology at liver of the fish, you will see the syncytial hepatitis or the syncytial giant cells in liver tissue. Sea bass is also the important species culture in Thailand and ASEAN country also. The important disease for marine culture, sea bass is including streptococcosis, Tennessee baculosis, scale drop, and muscle necrosis from Vibrio species, or you can call vibriosis, scale drop disease, virus infection, infectious spleen and kidney necrosis virus, ISKNDV infection, viral nervous necrosis, virus infection or VNN infection, iridovirus infection, and parasite. And for freshwater culture, Arsene sea bass, the disease will be similar with the tilapia because it's culture in freshwater also, such as streptococcosis, columnaris, aromonas, septicemia. The diseased fish usually infect by multiple organisms also in Arsene sea bass that you can call concurrent infection or mixed infection. The period or phase of culture Arsene sea bass will be similar to tilapia culture. This will be include hatchery, nursery, pre-grow out, and grow out period. And in all period of culture Arsene sea bass, you can see it can be infected with many kinds of pathogen, including bacteria and virus infection, and also other emerging disease. This is the columnaris disease in freshwater culture Arsene sea bass. Clinical signs similar to in tilapia also, including the gill necrosis and fin rot and tail rot. You may see the sudden back lesion, like you can see the white bar on dorsal fin of the fish also. Next disease is the Tennessee in marine culture sea bass caused by Tennessee maritimum. The clinical signs will be similar to Flavobacterium coronae infection or columnaris, but this bacteria will be infected in only marine fish because the bacteria survive only in marine water. Next disease is the disease caused by virus, scale drop disease virus or SDDV infection. According to the names of the virus we use, you can see the obviously clinical sign that is the scale drop. The scale is very easy to drop out from the skin of the fish, and you can see hemorrhage at skin of the fish. The next one is the scale drop also, but it's from the bacteria. We call this disease as the bacterial scale drop and muscle necrosis disease or SDMND. This picture is come from the natural diseased fish and the experimental diseased fish. You can see the same clinical signs that will be including scale drop and the muscle necrosis. Next disease is from virus also, infectious spleen and kidney necrosis virus or ISKNDV infection. When you see small fish infect with ISKNDV, you can see the obvious color change of the fish. Color of the fish will be black in color and pale gill. Fish will be like emaciation, or sometimes you can call like hemorrhage in the eyeball of the fish. Same as another iridovirus infection. You can find high mortality when the fish infect with iridovirus, and the eye of fish will got the hemorrhage at the retina of the fish. The streptococcosis you can find also in both freshwater culture and marine culture. Arsene sea bass. The clinical signs will be same as in tilapia. You can see the erratic swimming of Arsene sea bass infected with streptococcus. And also when you make a necropsy, you can see the hemorrhage in the internal organ and also around the fin and skin area of the fish. Okay, when we observe the farm and we found the fish infected with some pathogen, we may make a differential diagnosis what kind of the pathogen infect to the fish. When we investigate the diseases fish in farm and we make a differential diagnosis what kind of the pathogen infect that fish. In this talk, I will be focused on the molecular techniques and other advanced techniques that we have to use for disease diagnosis, pathogen diagnosis, and also antimicrobial resistance and antimicrobial susceptibility, how to do it. let's see on the next part. On this slide, I will start with how to deal with the diseased fish when you would like to make a diagnosis. First of all, you have to investigate the diseased fish in the farm. After that, only the clinical signs appear in the external area of the fish cannot make a definite diagnosis what kind of pathogen or what kind of disease that occur with fish. We have to make a necropsy and sample collection. The sample collection, you can keep it like fresh tissue, or you can keep in absolute alcohol fixation for another molecular diagnosis, such as the PCR later, or you can fix the tissue in formalin fixation for do another step with histopathology, or even you can use that tissue for in situ hybridization in the future. After you make a sample collection, you can do isolation and identification of the pathogen that will be include conventional techniques. Some pathogens are not convenient to do conventional techniques. You have to use immunological techniques also. And finally, some pathogen is very difficult and take time to use the conventional technique for identify what is the genus or species of the pathogen. You have to use molecular techniques sometimes. These molecular techniques and other advanced techniques, I listed the technique that we usually use in fish disease diagnosis is including the PCR, real-time PCR, sequencing, phylogenetics analysis after you have a sequencing, or you can use the bioinformatics to identify the pathogen or clustering the pathogen or characterize the pathogen. And also you can use bioinformatics for find antimicrobial resistant gene. That I will give you an example for the research work about that later. For diagnosis, you can use some advanced technique, not only PCR, such as loop-mediated isothermal amplification, or you can call LAMP, or RPA. If your lab have some advanced tools for identification and for antimicrobial susceptibility testing, such as Vitek2 system, you can use Vitek2 system also in some kind of pathogen, especially the pathogen in the group of Vibrio-Naceae or Enterobacteriaceae or Aeromonas. Anyway, some pathogen you still cannot use the Vitek2 system. You have to use another techniques. Next techniques that will use techniques of mass spectrophotometer for detect something that ionize from the cells of bacteria, the molecules that ionize from the cell of bacteria. After that, it will be interpret what kind of genus and species of pathogen. It will be very fast techniques for identify the pathogen. If you have it in your institute, but the multi-top machine is quite expensive. It's not important that you have to have all tools or all techniques in your lab if you would like to do the molecular research in the aquatic animal pathogen, or you would like to use molecular biology for study antimicrobial resistance. I think the best thing is you have to try to select the best method that appropriate to your institute, to your lab, to work with it. It's not necessary that you have to have multi-top. If you don't have multi-top or you don't have Vitek, you can use another techniques for identify the pathogen also. Sometimes the molecular techniques only cannot use for identify the aquatic animal pathogen. You have to use couple with the conventional technique or conventional method or another method also. I would like to say that the molecular techniques is not a magic bullet. Sometimes if you don't have it, you can use another techniques in your research also. Okay, but if you could work with it, okay, it's good. I cannot talk in detail in every molecular techniques that we have been used in study aquatic animal pathogen, but I think I will give you a case study in each kind of bacteria. First of all, I would like to give you a case study on the Flavobacterium infection. In my lab, we focus on the Flavobacterium as the first pathogen in our lab. Most disease laboratory studies are typically aimed at the single infection, but actually fish in the nature is exposed to various potential pathogens in the water. So that our hypothesis is the reality of death loss in culture fish farm due to multiple infection probably outweigh single infection. We have some outbreak in Thailand since many years ago, and we hypothesize like that it may not come from only one pathogen. We observe the mortality, the clinical signs, we collect the sample and do as the step as I told you before. We make a necropsy of each fish. We observe the clinical signs, both external and internal of fish. Then the next step, we have to do the bacterial isolation. For the bacterial isolation, it will be a little bit different in the pathogen of fish and aquatic animal because they have many kind of pathogen and all kind of pathogen not use the same spatial media for growing. For example, in the Flavobacterium columnar, if you would like to study with it, you have to use the selective media for them. You cannot culture it in the common media like nutrient agar or trypticate soil agar. You have to use the media called AOA. Or if you would like to isolate Francisella, you have to use cysteine hard agar for isolation of Francisella. But for other bacteria such as Aromonad or Edwardsella, you can use TSA for isolation also. It depends on what pathogen that you would like to study. After isolation, you may apply phenotypic and genotypic for identify the bacterial pathogen. In this case, we use the phenotypic or biochemical characterization, and we use 16S RNA sequencing for identify the bacterial species. Another pathogen, we use specific PCR diagnosis for internal organ of diseased fish, such as the specific PCR for Francisella, iridovirus, and Flavobacterium columnar. You can see that we cannot skip the step of the conventional method. Sometimes we have to start with it. Even finally, we will use the molecular biology or molecular tool for diagnosis or for identification. Okay, after we do the bacterial isolation and identification, in this case, we found a lot of kind of pathogen from diseased fish, not only a single pathogen, such as the Flavobacterium columnar, the Aromonas veronii, Biova veronii, Vibrio chlorella, Preciomonas chichteloides, Streptococcus agar-lactiae. And when we identify it by using 16S RNA sequencing, we can use the sequence that we obtain from the sequencing to use the bioinformatics to do the phylogenetics analysis. Then you can see that each kind of pathogen is clustering in or characterized in different group or different clade, including Aromonas veronii, Preciomonas chichteloides, Vibrio chlorella, Streptococcus agar-lactiae, Flavobacterium columnar. And when you use the specific PCR, you can see the difference in band pattern or molecular weight of each kind of pathogen, including Francisella, Flavobacterium columnar, iridovirus also found in this case. If we would like to know that all pathogen that we isolate from the diseases fish is contaminant or is reality pathogen for the fish, we have to use the Koch postulate to give back this pathogen to experimental fish, and the experimental fish should get the same clinical size, and we can isolate the same pathogen that we give to the fish from the diseased fish also. Okay, after we finish the experiment, we can found that the experimental diseased fish that infect with specific pathogen like F. columnar or Aeromonas vernii give the same clinical size that we found in natural diseased fish. The Francisella nortunensis subspecies orientalis also. Then we can conclude that the outbreak of the diseased fish in the area that we observe is not come from only single pathogen, but is from mixed infection or concurrent infection. Then we conclude that the reality of death loss in culture fish farm is due to multiple pathogen infection outweigh single infection. It answer our hypothesis, and after we make a conclusion and discussion, we write the manuscript and publish in aquaculture. And another kind of concurrent infection also published. This is the concurrent infections of Flavobacterium columnar and Edwardsella icteruli in striped catfish in Thailand. Okay, after our story, after we found the Flavobacterium columnar as a predominant infect to farm fish, we aim to study deeply into the virulence and characteristic of them. We found that colony of Flavobacterium columnar on specific media. We have two types, is rhizoid colony and non-rhizoid colony. Then we hypothesize that the rhizoid colony and non-rhizoid colony may make a different clinical size or virulence to the fish. Then we study more by collect the diseased fish sample. Then we isolate the bacteria, the Flavobacterium columnar from the fish, and we identification. You can see that finally we confirm by genotypic classification or molecular classification, but we start with phenotypic classification. I always say to my students that don't think that genotypic classification or molecular classification is the magic bullet. You cannot jump to that step without the phenotypic classification or the basic technique. So we should start with the basic technique of phenotypic classification first. We combine both technique, phenotype and genotype, for identify the Flavobacterium columnar. For the genetic characterization of the Flavobacterium columnar that we found, we use a few techniques for characterize it, including 16S RFLP, 16S rRNA sequencing RFLP, ISR sequencing and phylogenetics analysis, and 16S RNA sequencing and phylogenetics analysis that come from different reference. After we combine all techniques that we use together, we found the Flavobacterium columnar that we can isolate from tilapia in Thailand in different Genomova, mainly Genomova 2 and Genomova 1 in Thailand. And after that, we collaborate with Dr. Benjamin Laffen from USDA, and we publish this paper titled as Optimized Reverse Primer for 16S RFLP analysis, Genomova assignment of Flavobacterium columnar. In Thailand, as I said to you previously, after we do the phylogenetic tree based on ISR of Flavobacterium columnar, we can see that Flavobacterium columnar in Thailand can be divided in major is two branch 1 and branch 2. It appears unique in all techniques that we use for characterization. We compose all techniques together, and we propose that in Thailand we have Flavobacterium columnar that can be divided in different Genomova, Genomova 1 and Genomova 2. Sometime when you would like to characterization or differentiation of bacterial pathogen, you cannot use only a single choose or single gene for do the phylogenetics analysis or phylogenetic tree. You have to compose many gene or many techniques for characterize them. It depends on the discrimination power, it will be high or not. If you aim only single tools for do the characterization, you may fail. So that you have to try many tools or many methods for characterization. Then see the result can give you a high discrimination power or can characterize your pathogen very well or not. You can see it again after you try. As I said before, we have two colony morphotype of Flavobacterium columnar, rhizoids and non-rhizoid. We try to infect experimental fish with both rhizoid and non-rhizoid to see the virulence of both rhizoid and non-rhizoid. After we finish this experiment, we found that rhizoids has stronger in the virulence than non-rhizoid. Then we publish our result in Journal of Fish Disease in two titles: the phenotypic characterization and genetic diversity of Flavobacterium columnar isolate from lead tilapia in Thailand, and the virulent assay of rhizoid and non-rhizoid morphotype of Flavobacterium columnar. Okay, the last case study that I will talk today is how to use the molecular technique to study about the antimicrobial resistance or antimicrobial susceptibility in the bacterial species. This case study, I will give you the genome characterization of the quinolone resistant associated gene of Flavobacterium columnar, isolated from columnaris diseases ASEAN sea bass or sea bass that culture in fresh water. For the disease of ASEAN sea bass culture in Thailand, actually we found many diseases, as I give you in introduction part, that major infectious disease that we found, such as viral never necrosis or VNN, streptococcus infection, such as streptococcus ineae, scale drop disease virus or SDDV, and Vibrio species. And finally, Flavobacterium columnar is the disease that we found quite a lot in freshwater culture ASEAN sea bass in Thailand. Basic study about Flavobacterium columnar bacteria, it will be same as in tilapia. And also the clinical signs will be similar to tilapia also. The important that we would like to study in this case is the antimicrobial susceptibility and antimicrobial resistance. The human health hazard will be associated with the antimicrobial use in aquaculture also. The drug residues accumulation in aquaculture products and dissemination in environment, and rise of antimicrobial resistance or AMR. And the resistant gene transfer can be found. Several kinds of antibiotics have been permitted for use in Thai aquaculture, including amoxicillin, enrofloxacin, oxytetracycline, salafloxacin, oxalic acid, totasuryl, and many sulfur, dimetoprim, sulfur or metoprim drug. This is the antibiotic that we permit for using in Thailand, and it will be different in each country. It depends on the institute that control antibiotic using in each country. This study will focus on quinolone. The quinolone drug usually are restrict or prohibit in some country because of the critical importance of this class of antibiotics for treating human pathogen. But anyway, in some countries still allowed to use quinolone drug in aquaculture that you may have to concerns. because some kind of quinolone drugs also used for treating human disease. For antimicrobial resistance and monitoring, is also same case with the characterization of the pathogen, that even you aim in molecular study, but anyway you have to start with the conventional method. For Flavobacterium columnar, there are two most common methods for testing phenotypic antimicrobial susceptibility, including this diffusion method and broad or agar dilution method. We have a limitation for study drug susceptibility in Flavobacterium columnar. That is, Flavobacterium columnar are fastidious bacteria. You have to use the special media for them. If you use the common media, same as the bacteria in land animal and human, you cannot do the antimicrobial susceptibility test for Flavobacterium columnar. You have to use the special media. Because Flavobacterium columnar is usually clumping, so the protocol for AST, antimicrobial susceptibility testing, is quite complicated. It needs more standards method and need the official breakpoint criteria. For AMR gene, you can identify by PCR and DNA. For GIA and PASI in Flavobacterium columnar, we have been identified in Flavobacterium columnar before by Mata et al. 2018. We found a mutation associated with quinolone resistant phenotype were determined. The first study that revealed quinolone resistant strain harbor mid-sense mutation within the PAR-C and GIA, but not in GIA or PAR-E. This is the Flavobacterium, the data from Flavobacterium columnar that we isolate from tilapia, not sea bass. This is the first study of us that we identify the AMR gene by PCR. The limitation of this study is a single or few mediating resistant genes are tested. And if we would like to know the mutation, or if we would like to identify AMR gene in all strains that we have, it's a little bit expensive and time consuming to perform a complete identification of resistant gene. And some novel gene can be missing if we using the DNA sequencing technique for find the mutation or to find the AMR gene like this. So we can identify AMR gene by next generation sequencing, or we call NGS. It is the high throughput sequencing, can be rapid, it's become more feasible, and we can use the bioinformatics tool and free access database to analyze the sequence that we obtain from this next generation sequencing. The advantages by using next generation sequencing for identifying antimicrobial resistant gene is we can predict the antimicrobial resistant and antimicrobial associated gene. And we can tracking multi-drug resistant mechanisms by using some bioinformatic tools. We can investigate the horizontal gene transfer from next generation sequencing, and we can identify a novel resistant gene also. This is one example from another group of researchers that they identify the antimicrobial resistant gene from the complete genome sequencing, that by using next generation sequencing, they found around 286 genes were predicted as antimicrobial resistant gene when they blast to a card database. They found 32 genes were annotated as R&D type effect pumps, They found aminoglycosides and ganamycin resistant gene were identified. You can see that it's very feasible than you have to point or have to looking at gene by gene in the genome of them. But when you use the bioinformatics to analyze the whole genome sequence, you can find all antimicrobial resistant gene, or you can study about the mechanisms of antimicrobial resistance in the bacterial genome by using NGS. But anyway, for this research group, the systematical research of quinolone resistant mechanisms of Flawobacterium columnar still unavailable. That's why we aim to study deeply about the quinolone resistant mechanisms of our bacteria F. columnar that we isolate from ASEAN sea bass. And this is a case study. Our research plan is after we have F. columnar isolate from ASEAN sea bass, we do bacterial identification, phenotypic and molecular. We do antimicrobial susceptibility test by this diffusion and broad microdilution, and we compare both methods. Next from this, we do DNA extraction from the bacterial cells, and we submit the interesting strain to do whole genome sequencing or WGS by using NGS. And after that, we use the bioinformatics tools to analyze the resistome, or we can call the resistome analysis. The process of the research is we have to start with sample collection, of course, to find a lot of Flawobacterium columnar strain from ASEAN sea bass farm in Thailand. After that, we do phenotypic identification and molecular identification, same as we do in tilapia, Flawobacterium columnar. And after that, we do conventional diffusion method, Kirby-Bear test by using 7 kind of drug: oxytetracycline, amoxicillin, florphenicol, oxalic acid, nalidic acid, enrofloxacin, and ciprofloxacin. The special one for Flawobacterium columnar, the test should be performed by using 1.5 diluted Muller-Hinton agar or DMHA. You cannot use the normal Muller-Hinton agar because it will be very difficult to interpret antimicrobial susceptibility test for Flawobacterium columnar. And after that, inhibitory zone diameter were measured. After we found the pattern of antimicrobial susceptibility from this diffusion, we also do the MIC or minimal inhibitory concentration, and we focus on 3 kinds of quinolone and fluoroquinolone drug, including oxalic acid, nalidic acid, and enrofloxacin. Okay, after we interpret the antimicrobial susceptibility pattern, we will have the strain that sensitive to quinolone and resistant to quinolone. We select the strain from each group of susceptibility and resistant to do the next generation sequencing. After we obtain the data, we compare whole genome sequence for study about the resistome analysis. But before that, before we send for NGS, the DNA were extracted by Promega with such genomic DNA purification kit, and the DNA sequencing will be performed using Illumina high sequence platform. This one we didn't do ourselves in the laboratory. We submit to the company for sequence. They will expert more than us to do that. After that, we will go to the genome analysis workflow. After we got the raw data from NGS, we have to do contiguation. We have to do annotation by using rapid annotation using subsystem technology or RAS. And after that, we will use the comprehensive antibiotic resistant database or CARD to identify antibiotic resistant gene prediction. And we use the RAS finder to predict chromosomal mutation also. All of this is the bioinformatics tools to analyze the resistome, or you can call this workflow as the resistome analysis. This is the results of antimicrobial susceptibility of Flavobacterium columnar interpreted by this diffusion. We can see the interesting quinolone resistant strain, and we keep it at the interesting strain for whole genome sequence. We confirm minimal inhibitory concentration of each strain that we submit for whole genome sequence also. And we grouping as sensitive, intermediate, and the resistant or low MIC and high MIC before we submit for whole genome sequencing because we would like to compare the difference in each strain of bacteria that will be different in the pattern of susceptibility. And then 7 isolate of our study were selected for next generation sequencing. You can see that each isolate that we select has a different pattern of MIC value. Okay, this is the information of genome features of all 7 isolate that we submit for a sequence. And we found interesting antimicrobial resistant gene from CARD database. This is the overview of AMR gene by CARD. For quinolone sensitive group, we found the tetracycline resistant macrolide, fluoroquinolone peptide antibiotics, and glycopeptide antibiotics. For quinolone resistant group on the right hand side, total 173 genes were predicted. We found like macrolide resistant tetracycline, fluoroquinolone, glycopeptide antibiotic, and peptide antibiotic resistant also. For when we focus on quinolone resistant or QR associated gene, and we compare between quinolone sensitive group and quinolone resistant group, total 45 quinolone resistant associated genes were predicted. 40 genes were overlapped between quinolone sensitive and quinolone resistant group, and 5 genes were different, including these 3 genes and these 2 genes. Mostly the quinolone resistant associated gene that we found among our Flavobacterium columnar isolate is antibiotic effect pump resistant mechanisms and drug target alteration resistant mechanisms. You can see that a lot of bioinformatic tools you can use for identify many drug resistant genes in the genome of the bacteria, and also the mechanisms. The bioinformatic tools will help you to easily finding antimicrobial resistant gene and mechanisms and grouping them. From our result from RAS Finder program or RAS Finder bioinformatics tool, we found 13 chromosomal mutation were predicted from this RAS Finder, including this one. And the gene that I make a marker, this one is the gene associated with quinolone or fluoroquinolone resistant in the genome of the bacteria. And after that, we identify the mutation in this gene by MECA7 software. This is our conclusion after we study whole genome sequencing of the bacteria. We can overview of quinolone resistant mechanisms of Flowobacterium columnar that is including the resistant quinolone resistant in Flowobacterium columnar is including the target size gene mutation and many genes related to the effect pump. In the conclusion, most of Flowobacterium columnar isolate were resistant to the first generation of quinolone, such as oxalic acid and magnesic acid. And the main quinolone resistant mechanisms of Flowobacterium columnar are target size gene mutation followed by effect pump activity. Major type of effect pump associated with quinolone resistance is belong to RND effect pump family. The novel mutation in JB and PAR-C were discovered in our study. Then the further research of proteomic may be needed in the further study. Okay. The result and conclusion obtained from our study was published in aquaculture in the title Resistome Characterization of Flowobacterium columnar isolated from fresh water culture ASEAN sea bass reveal diversity of quinolone resistant associated gene that just recently published in 2021. Okay, that is all for my talk and give you a case study. My talk will give you some idea that if you would like to use the molecular techniques for study aquatic animal pathogen and also monitoring the antimicrobial resistance in aquaculture. Thank you very much for your attention, and I would like to end with acknowledgement of our research team. Dr. Ha Tan Dong, Dr. Pathanapol Khayan Samruat, Dr. Senapin, Dr. Nopadon Piralat, Dr. Benjamin Lafent, and also my student in Center of Excellence of Fish Infectious Disease. Okay. This is our activity in aquaculture in Thailand. Thank you very much for your attention.
Yes, thank you very much, Dr. Channarong Rodkhum, for incredible speech. You already have many research and published in the top journal of aquaculture, right? So, Dr. Let me remind you, Dr. Channarong already talked about molecular research in Thailand's aquaculture, pathogen characterization, phenotypically and also genotypically, and also antimicrobial resistant monitoring by using the next generation sequencing. So very interesting. I believe you have already many discussion, question, and also comments. So you can put your writing, your comment, your question in a pigeonhole that already shared in chat room. So that next is a discussion time. So I invite Dr. Channarong Rodkhum and also Professor Wayan to open your video. Professor Wayan, are you here already? Yeah, Mbak Putu, maybe you can show the question from the audience.
Okay, doctor, please wait for a while.
Okay. So this is for who, Mbak? For Prof. Wayan or for? Okay, first maybe for Dr. Channarong Rodkhum from Budias Nursanti. Is there any report of AMR case in non-aquaculture practice? If it is, how to control it? So you can directly answer the question, Dr. Channarong.
Okay, thank you very much for the question. I think you mean not only in aquaculture practice. Are there any AMR case? Yes, I will say yes, of course. They have AMR in all area of animal culture, not only in aquaculture, in livestock also. Yes, how to control it is quite long story if I have to say about the policy to control it. But if I will say briefly in this seminar, I will say that we have to cooperation in all sector, including the farmer, veterinarian, and the company that's selling the antibiotics to the farmer and or to veterinarian. We have to make a prudent use of antibiotics in the area of agriculture, both aquaculture and livestock. And we have to educate farmer how to deal with the antibiotic use in aquaculture and in livestock. For example, they have to know about how long they have to use, how long they have to stop using the antibiotic drug before selling the agriculture product or aquaculture product to the market. And I think lastly, last but not least, is they have to like concern using antibiotics a lot. And if they have the alternative way to avoid using antibiotics, we should promote them to use, such as using the probiotics or immunostimulant for promote the immune status of animal before the disease will happen in agriculture or in aquaculture. Yeah, I will answer like that. Thank you very much.
Yes, thank you, Dr. Chana. So I think we agree that cross-sectoral collaboration to tackle this problem will be very powerful, right? Then the next question for Prof. Chana, maybe. Prof. Wayan maybe still have problem with the connection. Maybe we can, yeah, maybe first for the for Professor Chana, maybe. Yeah, how to from the same audience, Putias Nursanti, how to detect pathogen of a new emerging disease in the outbreak situation while we still have less information about the infectious agent, and when it is RDT developed, how to measure its accuracy. Please, Dr. Channarong.
Okay, thank you very much for the question. I think a lot of students may ask about this also when they would like to study about emerging disease, or especially the virus that we never been found in agriculture or in aquaculture before. I think first of all, we have to have a standard way to investigate the disease. As I said, we have to observe the clinical signs. We have to make a necropsy if in animal, and after that, you may find some new emerging viral disease, especially the viral disease in the histopathology, or you may find in the evidence of it can be emerging disease in some step of your study. Yeah, I see some emerging virus in tilapia from that step also. Before we investigate the disease or we observe the disease, we never know that we will find that iridovirus in tilapia before. or we never found the tilapia lake virus in tilapia before. But after we see the histopathology, we just see in the histopathology, then we found that some inclusion body is in the histology cells. Then we use we have to amplify the virus by using the universal primer for virus or even for bacteria. Then we scope down what kind of bacteria or what kind of virus we found in that emerging disease. Then you may apply with the another molecular techniques to identify it. Finally, you will get the new emerging disease. Yeah.
Yes, thank you, Dr. Channarong. So first from clinical sign, right, and then go deeply to the for example, histopathology, and then PCR, and so on and so on.
Yes, Dr. Dini, I will say again that we cannot skip the step of the conventional. We have to start from disease investigation and diagnosis. Then in between the step, you will find something that emerging disease. Yeah.
Yes, thank you so much for the answer. And next. Prof. Wayan already here, right, Prof? Hello, Prof. Hello, Prof. Wayan. Can you hear me? So for Prof. Wayan from MSAR, so I would like to know, does the molecular identification or detection will be used for all zoonotic diseases in order to detect diseases at early stage? Can you hear me, Prof. Wayan? Or maybe Dr. Channarong Rodkhum, could you please answer this same question?
Okay. Yes, of course, you can use the molecular identification and detection for zoonotic disease. Is also you have to base on the clinical signs that you observe from the animals. Let's say that you have to have a basic background about the disease in animal before. I will give you an example for melioidosis and tuberculosis in animal. After if you be a veterinarian, you know the clinical signs of animal that got the disease of the melioidosis or tuberculosis. Then you have the first step, you have the differential diagnosis. What is this disease? 1, 2, 3. After that, you have to rule out 1 or 2 or 3 is correct by apply the conventional techniques or molecular techniques to identify and detection and rule out it one by one. Melioidosis or not, tuberculosis or not, yes, or nocardia or not, then you will get the final identification. I'm not sure is my answer is will be right to the question that you asked or not. Yeah.
Yes, thank you so much. Principally, absolutely yes, you can use molecular identification for any zoonotic diseases, right? Yes, thank you.
Yes,
Dr.
of course, yes.
Channarong. And then next question. Next question is also for Prof. Chana from Muhammad Aji Ikrami. How do we trace the distribution of ARG to the environment through water, aquaculture to environment, as we know that the movement of water is very dynamic? Time is yours, Prof. Chana.
Yes, thank you, Dr. Dini. of course, we can do it. We can trace the distribution of antimicrobial resistant gene in environment and water. Right now, we have like the techniques that we extract environmental DNA or the DNA from the living organisms in water. We can concentrate it and we extract it, and then we can use that one to study about microbiomes or antimicrobial resistant gene in the DNA that we extract from environment and in the water also. Yeah, of course, yes, we can do it. Yeah, same step as the bacteria that we isolate we isolated from animal tissue also. Yeah.
Yes, thank you so much, Prof. Chana. So you can use environmental DNA technique, right? eDNA, yeah, to take a water sample and also sediment sample from the movement water, also from river or also from lake, pool, and so on. Thank you. So next question. So yes, the next question is from Research Station Office Health Control. Why different clinical sign between Aeromonas hydrophila and Veronii, Aeromonas veronii? What is the different clinical sign between Aeromonas hydrophila and Aeromonas veronii? If in one case you found many pathogen, how we know we have the role as primer or secondary infection?
Okay.
Is it
okay,
primer?
thank you very much for this question. It's very good question, and is a problem for identify these two bacteria from the diseases fish in many countries also, since Aeromonas hydrophila and Aeromonas veronii is very closely related. It's very difficult to using conventional technique. This is one of the most case that is very difficult for using conventional technique. You may have to use the molecular technique for identify it. I will say that in case of Aeromonas hydrophila and Aeromonas veronii also, you have to use the molecular such as 16S, RNA, RPO, D gene, or another gene that they have the discrimination power to discriminate between Aeromonas hydrophila and Aeromonas veronii. If you ask about the clinical side, I will say from my experience that it's really hard to differentiate the bacteria, the disease that infect by these two bacteria by clinical signs. The clinical signs look very similar, including the patchy hemorrhage or the large area of hemorrhage around body surface and fin, and also the internal organ of the fish. Yeah, but we may make a differential diagnosis based on epidemiological data in each country. For example, in Thailand, in tilapia disease, we usually found Aeromonas veronii and some Aeromonas schubertii and Aeromonas dickensis in tilapia more than A. hydrophila. Yeah, you may base on epidemiological data in your area also, but for the clinical side, it's hardly to differentiate it. Yeah.
Yes, thank you, because almost same, then the clinical sign is very difficult, so you can use epidemiology sign from before cases, right?
Yes.
Thank you.
Yes.
So next question, Mbak Putu.
Bu Dini, saya bisa pakai HP.
Oh yes, thank you, Prof. Wayan. So from
okay.
Yes. From the question is from Maharani, Prof. Wayan. In Indonesia, what is method to studying the human microbiome? It is it is need or not ethical clearance if we will conduct this research? Thank you. From Maharani, Prof, please.
Yeah, okay, thank you. Every research, we need ethical clearance. If you work with animals or in human, we need ethical clearance. Of course, before we done our research, to do our research, we should have an ethical clearance to do research in the field as well as in the population. So for microbiome analysis, we should have a molecular technique that we explained by Dr. Channarong. New microbial detection could be not possible to use in a conventional one. We should use a molecular technique to do that.
Yes.
Okay, that's yeah.
That's all, Professor?
Yeah.
Oh, yes.
Yeah.
Thank you so much. Thank you so much for the answer. And next question, maybe. Are there still any question from the audience? Or also for Prof. Wayan, based on zoonotic diseases, does Indohun or UGM conduct research or surveillance collaboration for the detection of COVID-19 in animals around human cases? From Ati Ratnawati. Please, Prof. Do you have any?
Just
Yes.
Thank you so much. We did COVID-19 in collaboration with Indohun. We have 14 provinces to map the COVID-19 in a high risk province, 14 province. But in animal, we just doing for research in a few, not all animal. Just we try now. We have one grant that we will identify COVID-19 in also in animal, but not yet run for today. For human, we did already.
Yes, we did already for the human. For the animal, still on progress.
Yeah.
I mean, in the future. So
we map potential the disease transmission from one population to another population, and also mapping the COVID-19 in the Kulon Progo district. And we do also active surveillance using antigen detection.
Yes.
Okay, thank you.
Yes, thank you for the answer, Professor Wayan. So for the next question, are there still any questions from the audience? Yes, from Aji maybe for Professor Channarong. Is there any possibility of zoonosis originating from the aquatic environment? Because the condition of fish, cold blood, and human are different. Please, time is yours.
Yeah. Actually, thank you very much for this question. I would like to say also after my talk that we also have some zoonotic pathogen that can be infect human also, and is cause many disease in human already right nowadays. That one such as the streptococcus. Some clone of Streptococcus agaricale that can be cause the septicemia in human, and in fish they also have the mycobacteriosis caused by Mycobacterium malinum, Mycobacterium fortuitum, and Mycobacterium chelonae that can be infect human also. And the human case that we found infect with that tuberculosis is usually the immunocompromised human that stay together with the activities that incorporate to the aquarium, such as aquarium watching, or like the immunocompromised patients that work with fish farm. They can infect with some zoonotic pathogen also. Yeah, even Aromonas species can be infected to immunocompromised patients also. Yeah, and is still a lot of zoonotic pathogen actually that can be infected from fish to human. Yeah.
Yes, thank you, Professor Chana. At least Mycobacterium and Streptococcus already reported that also infect human being.
Yes,
so thank
Dr.
you.
Dini.
Yes, thank you. Next question, maybe. Are there still any questions? Yes, this is from Research Station of Fish Health Control. So maybe for Prof. Wayan, is One Health in the Hoon only? Oh, maybe both of you, for both of you. Is One Health in the Hoon only collaboration with universities? How about collaboration with research institutions? What is the requirement if we would like to join with Indohoon? Thank you. Maybe Prof. Wayan will answer this question first.
Yeah. Yeah, thank you. This is a good question. I think because One Health Indohoon, that the definition, the meaning of Indohoon is Indonesian One Health University Network. Still, the network among the university member, we have 34 faculties members around Indonesia from 20 universities. But we want to extend the collaboration in the departmental and also in institution, but not belong to the Indohoon, because Indohoon is to university network. And then this is One Health network, not Indohoon. But we have already collaboration with Department Pertanian, Agriculture, and the Department of Health, and also many different institutions outside universities. So, yeah, this is my question, my answer. Thank you.
Yes, yes, thank you, Prof. Wayan. Maybe Prof. Chana will add some more information about it.
Yeah. Actually, I am not an organizers in Sihun or Chohun in Thailand. Yeah, but as I know a little bit, even you're not in the university, you are not the university researcher or not a student in university, you can join the Sihun, Indohoon or Chohun for Thailand also. Yes. I think the Yes. And I think Sihun has organized the many One Health organized in each country already. Yeah, we have in Indonesia, right? In Philippines, in Thailand. Yeah, in I think many country in ASEAN country already have some HUN or some SUN already. Then the people, the researcher or the student in each country can join the CHUN by passing the institute in your country. Yes.
Yes, thank you. Thank you, Prof. Chana. Are there any?
Budini, just a little bit information, Budini.
Yes, okay.
For those that not yet involved in the One Health network, Sihun is offer in national regional network. There's also offer some competition for the student and also for One Health interest people. And then in each country they have we have already like Indonesia, we have Indohoon, Malaysia, Myhun, and Thailand Tohun, and also Myanmar, the Philippines, Cambodia, all Asian countries have established One Health network. Yeah. Besides One Health network, there are also a GRIS network in regional Southeast Asia. Okay.
Yes, thank you, Prof. Wayan. So One Health already have many collaboration in many countries. I think this is very important. So are there still any other question, Mbak Putu, from the audience? Mbak Putu?
Yeah. I think no question more, Budini. So I think we can finish for the Q&A session.
Okay. Yes, thank you so much for the participation, active participation, and also a very fruitful discussion. And thank you so much for Prof. Wayan and also Prof. Channarong Rodkhum for the very nice talk about the application of molecular technology in zoonotic diseases, both from terrestrial animal and also from aquaculture, right? And the importance of identification for human risk reduction. So thank you so much for the very nice talk. So I return back to the MC. Time is yours. Thank you.
Thank you very much, Dr. Dini. We have a very
Thank you,
interesting
Bu
discussion
Dini.
and lecturing. And as that you already just mentioned to us that we learned a lot about the molecular technology, the importance of the rapid diagnostic, which is a product of molecular technology in controlling zoonotic disease, and the application of this technology in aquaculture, in investigating fish disease, and also antimicrobial resistance. This is very interesting and awesome topic today. And then before I end up this session, and I know all of you already asking about the attendance form. I'm sorry. I know that some of you already asking the attendance form, but please, can I have all of your picture? So turn on your video because I will take a picture with all of you. So Dr. Channarong and Prof. Wayan, would you please to turn on the video and all of the participants? And after this, I promise to send you the attendance form to all of you. Today we have a lot of participants, not only in Indonesia, but we have also in Vietnam, Thailand, Philippines, and some of from Malaysia. There are a lot of people coming from abroad. Okay, so I will start to take a picture. Oh, wait a minute. Oh, wait. Okay. Okay, 1, 2, and 3. Once again, 1, 2, and 3. Keep your smiling because you don't know which screen are you located on. 1, 2, and 3. The next screen, 1, 2, and 3. The next screen. Okay, the last screen. 1, 2, and 3. Thank you very much. I will share you a link, and maybe I will ask my friend to share the screen in the chat rooms, and I will share you. You can just scan in the by your phone on this barcoding. And lastly, but not least, I would like to thank you again for our awesome speakers. Thank you very much, Prof. Wayan, and thank you very much, Achan Channarong, my advisor in Chulalongkorn University. Thank you very much for making it happen, Achan. And also thank you for Dr. Dini for leading the session. And then thank you very much again for all of the participants that coming today. We will share the e-certificate after one week, and also the material from the speakers after I get the permission from them. Thank you very much and have a good day. Bye-bye.